Hello everyone, welcome back to the class. In previous class we learnt about how the transport of protein happens through a cistern. There were two models if you remember, which explained the transport of proteins through Golgi cistern. That is, one is cisternal maturation model, and second one is vesicular transport model. According to cisternal maturation model, stacks of cisternae were dynamic. Cismost cisternae is assembled by the fusion of transport vesicles from the ER carrying newly synthesized cargo. Each cargo cisternae matures as it migrates outward through the stack. And at each stage, the Golgi resident proteins that are carried forward in the cisternae are moved backward to an earlier compartment. Each cisternae progresses into this next one, and COP1 coated vesicles functions to transport resident Golgi components in the retrograde direction. The other model which explained the transport of proteins through cisternae was vesicular transport model. According to this, a stable stack of cisternae containing a fixed and ordered array of the processing enzymes. Cargo is delivered to each cisternae in turn by vesicles that bud off from one cisternae and fuse with the next in the stack. In this case, COP1 coated vesicles will follow the anterograde direction movement to transport the particular protein across the cisternae. Next, we learnt transport of proteins proteins from the TGN to lysosome. Which are the steps? Let us recall. First, it was addition of phosphate to mannose. The particular carbon in which the phosphate was attached, that is carbon 6. So we call it as M6P. What is M6P? M6P is a oligosaccharide which is attached to the protein of our interest. After this there is removal of N-acetylglucosamine, that we can say, I'll write it like this: removal of N-acetylglucosamine. And after that there is M6P receptors. Where are these present? M6P receptors, where are these present? They are found in the trans-Golgi network. The M6P of the protein will bind to the M6P receptor, which is present inside the trans-Golgi network, where not only this, there is a vesicle, transport vesicle, which is a clathrine AP1 coated transport protein, inside which there is MP6 receptor, to which the M6P receptor is present, to which M6P of protein has attached. This unit will fuse with the sorting vesicle in the late endosome, which has an internal pH of approximately 5.5. The bound lysosome enzymes are released within the late endosome. That's what we learnt. See, the addition of initial processing of the preformed N-linked oligosaccharide precursor in the rough ER is same for lysosomal enzyme. In cis Golgi, one or more mannose residue in the resulting manoseate N-acetylglucosamine oligosaccharides of lysosome enzymes becomes phosphorylated. From where this phosphate has been inserted to mannose, it is from phosphorylated N-acetylglucosamine. Where this phosphorylation is happening, carbon 6 of mannose. The enzyme phosphodiesterase removes N-acetylglucosamine after phosphorylating mannose. This M6P binds to mannose-6-phosphate receptor, which are primarily found in trans Golgi. The luminal domain of this transmembrane protein contains a region that binds M6P very tightly. Clathrin or AP1 coated transport vesicle containing M6P receptor are bound lysosome bud off from the trans Golgi network. And it is transported to the endosome, where the bound lysosomal enzymes are released within the late endosomes. What happens in lysosome after there is fusion? Phosphatase within late endosomes removes the phosphate, and it causes preventing their rebinding to the M6P receptor. This results in two types of vesicle budding. That is one type which is having lysosomal enzyme, but there is no M6P receptor. Other type of vesicle is coated with retromer coat. After these vesicles bud from late endosomes, they fuse with lysosome, delivers the lysosomal enzymes to their final destination. Then what is the fate of receptor? It gets back to the trans Golgi network. This is the process we learnt yesterday. And this table will remind you the signal sequence, specific signal sequence, its function, and the type of vesicle used to transport. Today we are into vesicle fusion. What is vesicle fusion? The fusion of transport vesicles with its target. That involves two types of events. First event is transport vesicles that must specifically recognize. Consider there is a transport vesicle. It is a transport vesicle. If it is to fuse, it should recognize the target membrane. After recognizing, that will fuse with the membrane, target membrane. Let us see. Vesicle fusion mediated by specific recognition between a vesicle and its target by interaction between unique pairs of transmembrane proteins. Fusion between the phospholipid bilayers of the vesicle and the target membrane happens. See, there is an image I have to show. See, this is the cytosol. This is lumen. It is a type of membrane where there is a transport vesicle budding off. This can be trans Golgi network, from where the budding off of transport vesicle is happening. There is budding off, and it is recognized, and this transport vesicle recognizes the target membrane to which it is destined, and there is fusion. The contents inside the transport vesicle gets into the target compartment. So there is a particular steps or the hypothesis to explain this vesicle fusion. So that is SNARE hypothesis. What? The hypothesis is called what? SNARE hypothesis. What does it explains? It explains the vesicle fusion with target. There is a particular concept in the SNARE hypothesis, that the vesicle binds or fuses with the target membrane with the help of with the help of interaction between pairs of or pair of proteins. What does this hypothesis say? The vesicle fuses with the target with the help of interaction between pair of proteins. What are these pair of proteins? These pair of proteins are called SNARE. That's why the hypothesis is known as SNARE. What is the full form of this SNARE? The protein interaction between pair of proteins, that protein is known to be SNARE. SNARE, full form of SNARE is SNAP receptor. SNAP receptors, SN and SNA and RE. Therefore, the name is SNARE. So what is the pair of proteins called? SNARE. What is the full form of SNARE? SNAP receptor. So these SNAP receptors or SNARE proteins are present in both vesicles and target membrane. They are present in both vesicles and target membrane. The SNARE protein or the SNAP receptor which is present in the vesicle is known as Vesicle. I'll write it in a different color. So the SNARE protein or the SNAP receptor which is present in the vesicle is known as Vesicle. The SNARE protein which is present in the target membrane is known to be T-SNARE. So what did we learn now? The SNARE hypothesis explains how the vesicle fuses with the target, and that is mediated by interaction between specific pairs of proteins. That proteins is known as SNARE, which is known as SNAP receptor. That SNAP receptors are present in both vesicles and target membranes. They are known to be V-SNAREs and T-SNAREs. The V-SNAREs, in vesicles there is V-SNARE. I'll write it like this, the larger one. The vesicle is having V-SNARE and the target membrane is having T-SNARE. The interaction between these two proteins will lead to the fusion. That is according to the SNARE hypothesis. In neuron, there is a particular V-SNARE and T-SNARE, and there is a particular name for this V-SNARE and T-SNARE. The V-SNARE which is present in the synaptic vesicle is known as synaptobrevin, which is also called VAMP, V-A-M-P. In neuron, the V-SNARE which is present in the synaptic vesicle of the neuron is known as synaptobrevin, that is VAMP. And also the T-SNARE is also present. T-SNARE of neuron has a particular name, that is syntaxin. Syntaxin is also called as SNAP25. So it is the name of particular T-SNARE and V-SNARE which is present in the neuron. So in neuron, what is the V-SNARE called? V-SNARE is called synaptobrevin. Synaptobrevin. This synaptobrevin is also called VAMP, V-A-M-P. And there is T-SNARE also. T-SNARE is called what? Syntaxin. syntaxin, that is also called SNAP25. Both of these V snare and T snare forms a bond, or they fuses with each other, and they result in transmission of particular signals or neurotransmitters. Okay, the speciality of the syntaxin is that it is lipid anchored protein. It is lipid anchored protein. It is lipid anchored protein. It is attached to the membrane by palmitylation of 4-cysteine residue. So it is lipid anchored protein. So we learnt the classification of snare based on its location, that is V snare, which is present in the vesicle, and T snare, which is present in the target membrane. There is another classification based on identity of highly conserved residue, that is, this is the second classification. Of snare, that is R snare and Q snare. Now what is this R snare and Q snare? R snare is a arginine containing snare. Q snare is glutamine containing snare. So this classification is based on what? Based on identity of the highly conserved residue. So R snare is made up of arginine, Q snare is made up of glutamine. Remember this. So let us now see one by one. See, this model explains vesicle fusion with target is mediated by interactions between specific pairs of proteins called snares, that is SNAP receptor, on the vesicle and target membrane, V snare and T snare. V snares pairs with cognate T snares in the target membrane. In neurons, V snare is called synaptobrevin, which is vesicle associated membrane protein, that is full form of VAMP, and T snare are designated as syntaxin, that is SNAP25, that is 25 kilodalton synaptosome associated protein. Both syntaxin and VAMP are anchored to the membrane by a carboxyl terminal of the transmembrane domain. SNAP25 is a lipid anchored protein attached to the membrane by palmitylation of 4-cysteine. Snares have also been reclassified based on the identity of a highly conserved residue, that is R snare made up of arginine, Q snare made up of glutamine. Now let us see the explanation or the practicality of this hypothesis. Recognition step is controlled by Rab protein. So I'll explain it in a raw method. So the interaction between V snare and T snare is mediated by a protein called Rab. The interaction between V snare and T snare, I'll write it like this. The interaction between V snare and T snare is mediated or checked by Rab protein. What protein? Rab protein. Okay. This Rab protein is of two types, that is GDP bound state and GTP bound state. The speciality of Rab protein in GDP bound state is that it is inactive. And remain in cytosol. The Rab protein in GTP bound state are active and tightly bound, tightly associated with the membrane of an organelle or a transport vesicles. So what makes this Rab protein to GDP or GTP? You remember You remember I had given a term earlier that was Jeff. Do you remember this? Jeff. Guanine nucleotide exchange factor, which donates the guanine. The GDP is converted to GTP by this factor. When GEF reacts, or the membrane bound GEF activates the RAP protein by giving out guanine, making it GTP. It becomes active. So the activated RAP protein binds with the RAB effector. Consider now, RAB protein is inactive. RAP protein is inactive. It is activated by what? It is activated by Jeff giving what? Guanine. So now there is activated RAB protein. This activated RAB protein reacts with or binds with RAB effector. This RAB effector is nothing but a tethering protein. So RAB effector proteins interact with RAB proteins located on the target membrane, vesicle membrane, or both. So this RAB effector reacts with both RAB proteins present in V-snare and vesicle and target membrane. So there is interaction between this. This leads to the binding of V-snare and T-snare. Okay, after that, after the interaction, after the binding of V-snare and T-snare, so this results in fusion of V-snare and T-snare, as I said, right? After this, to remain bound for long enough to allow the RAP protein to hydrolyze its bound GTP. So RAP protein, after fusion, after binding of V-snare and T-snare, RAP protein hydrolyzes its bound GTP, which locks the vesicle onto the target membrane. So after this, what happens? After this, RAB protein hydrolyzes GTP. RAB protein hydrolyzes GTP. Okay, after this hydrolysis, so it locks the vesicle onto the target membrane. After tethering, snares mediate docking and fusion. So now is docking and fusion. What happens in this step? Let us see. The complex have to disassemble before snares can mediate new round of transport. So consider the V snare has come in contact to the T snare, and now it has to disassemble so that it can release the protein which is present inside that vesicle to the target membrane. After V snares and T snares have mediated the fusion of vesicle on a target membrane, the NSP, there is NSP, that is what? NEM sensitive factor. NSP will binds to the snare complex, via adapter protein. Sorry, this is NSF. NEM sensitive factor. NSF is a NEM sensitive factor. NSF is a hexamer of identical subunits, and snaps are not necessary for actual membrane fusion, but rather are required for regeneration of free proteins. NSF is soluble ATPase and hydrolyzes ATP to dissociate the snares apart. So let us now see the actual process. Recognition steps is controlled by Rab protein, which checks the interaction between V snare and T snare. This is V snare. The red one is the V snare. The blue one is T snare. The recognition of this both is mediated by what? Rab proteins. Rab proteins. Rab protein in GDP bound state, they are inactive. In GTP bound state, they are active and are tightly associated with the membrane of an organelle or transport vesicles. Membrane bound Rab GEFs activate the Rab protein. So Rab GTP is in active form now. Once GTP bound and membrane bound state, Rab proteins bind to other proteins. So this Rab GTP will bind to this Rab effector, which acts as a tethering protein. Rab effector proteins interact with the Rab proteins located in the vesicle and in the target membrane. The binding of V snare and T snare. So the binding of V snare, the red one, and T snare is happening here. To remain bound for long enough to allow the Rab protein to hydrolyze its bound GTP, which locks the vesicle onto the target membrane. So here you can see hydrolysis of Rab GTP happening. After tethering, snares mediate docking and fusion. Interacting snares need to separate apart before they can function again. They again get separated to make the process repeatable. Most snare proteins in cells participate in multiple rounds of vesicular transport. After that, what happens? After there is fusion happening, the NSF, this is NSF, NEM sensitive factor, binds to the snare complex via adapter proteins, that is SNAP proteins, soluble NSF attachment proteins. NSF are soluble ATPase and hydrolyzes ATP to dissociate the snares apart. So this is the model how V snare and T snare gets in contact with each other, and how the fusion takes place between the vesicle and the target membrane is shown in this image. So this is the whole thing, where from starting from hydrolysis or the activation of Rab protein to the fusion, this is the thing, how the transport of proteins from the trans Golgi to the lysosome, or what the snare hypothesis says. See, the NSF binds after the V snare and T snare have mediated the fusion of a transport vesicle with the target membrane. NSF binds to the snare. NSF binds to the snare complex, and with the help of accessory proteins, it hydrolyzes ATP to make the snares apart, to dissociate the snares apart, so that the cycle can run again. This is the another image depicting the fusion and snare hypothesis. See, this is the cycle. So in next class we will be learning about lysosome. Thank you.